we have one more panel to discuss Rising Stars of Neuroscience in Texas, and it's going to be moderated by Daniel Johnson, professor in the Department of Neuroscience at the University of Texas at Austin. Dr. Johnson is also a member of our 2019 Planning Committee, and so we're very grateful for him organizing this panel. So thank you so much.
So I'm really pleased. It's a pleasure to moderate this session on rising stars of Texas. Fortunately, Texas has a lot of them. We've already heard from quite a few in this meeting, and we have the good fortune to hear from 3 more. So we're going to hear about brain circuits and how they function, brain rhythms, and the role they play in brain function, and also the structure-function relationships of a particularly class of proteins, neurotransmitter receptors, which are important for binding the chemicals. So we have Brian Arenkiel, Benjamin Arenkiel, Laura Lee Colgan, and Ryan Hibbs. So let me first introduce Benjamin Arenkiel. He's associate professor at Baylor College of Medicine in the departments of molecular and human genetics, Department of Neuroscience, and in the program of developmental biology. He got his degree in microbiology and chemistry from St. Cloud University. He did his doctoral degree in genetics from the University of Utah, where he trained under Nobel laureate Dr. Mario Capecci. He then did postdoctoral work with the late Larry Katz and Michael Ehlers at Duke University, and then joined Baylor College of Medicine as the first McNair scholar. So Dr. Arenkiel is currently investigating how genes and activity interface to build and maintain, remodel neuronal connections in the brain, as well as research in the area of basal forebrain, and circuits that influence eating and addiction behavior. So welcome, Dr. Arenkiel.
Okay, so we're kind of on the home stretch here. I want to thank the organizers, in particular Joe Takahashi, for the invitation. It's a really thoughtful invitation and distinguished group of in the audience. So what I thought I'd do today is talk a little bit about a fairly new line of research in our lab that has emerged from really a serendipitous series of observations that I think what we're learning is that we've uncovered some discrete brain circuits that take sensory information from the environment and directly pipeline it into some pretty overt physiologies. And in particular, I'm going to tell you about one circuit that alters dramatically feeding and addiction behaviors. So to start the story, I'm going to consider, first of all, our primary line of research, which is really focused on understanding the mechanisms at the cellular, genetic, and synaptic level of how you build brain circuits. And this has really guided our research from the beginning. But really, this is an oversimplification, but really, what we're trying to get at here is that we're working under the assumption that all of the bad things that happen to brain tissue are from dysfunctional brain circuits. So our lofty goal is to come up with ways, genetic and cellular approaches to fix brain tissue, but even probably more proximally, understand a little bit better the patterns of connectivity as well as plasticity mechanisms to perhaps take advantage of the complexity of the brain and reroute information in a way that we can perhaps ameliorate some of the bad things that go wrong with the brain. So clearly this is a pretty lofty goal. So our agenda is to embrace the mouse as our model system. And this is really because we have a lot of genetic power. We're able to really mark, manipulate, and monitor brain circuits and cells really at will right now. The only thing truly limiting us is our own imagination. And in fact, we embrace a system within the system. Shown here, pseudo-colored in yellow, is the mouse olfactory system. And the reason why we like the system is multifold. First of all, it's directly related to behavior. So if you're going to study brain, it's nice to have an inroad into an actual behavior. It's experimentally tractable. It's anatomically defined, pretty simple compared to other sensory systems, but more importantly, we embrace this interesting feature of ongoing adult neurogenesis. So this is one brain region alongside the hippocampus that continually supplies new neurons to the brain, so we can use this as a substrate to understand how, in particular, we might be able to rewire adult brain tissue. So we've been spending a lot of time into this in the last few years, is understanding how to rewire new adult-born neurons in the olfactory system. But along the way, we've been captivated by this interesting concept that we're all familiar with, is certain types of physiological state are known to kind of increase our brain health and brain plasticity. So we all have heard learning, brain games, even exercise can promote brain health and plasticity. So we've been kind of a long-term agenda to try to find the molecular correlates of how this type of physiology can actually transmit at the molecular and genetic level into a better brain circuit. So I'm not going to go into the details of how we found this, but we've, in short, we've anatomically identified a really nice candidate brain region that might be doing this. This is the deep basal forebrain. You can see here in green, there's green projections projecting all the way up into the olfactory bulb, and they make specific synaptic connections onto these adult-born neurons. And we took a particular interest in this brain regions because it's populated by a subset of neurons that secrete acetylcholine. Acetylcholine was intriguing to us because we know a lot about acetylcholine as a neurotransmitter. It's known to influence olfaction, so it fits into our circuit. It's synaptogenic in vitro. It can serve as a neuronal stimulant. It's been linked to reward and arousal, so these different types of physiological states. And interestingly, mouse models of Alzheimer's disease treated with acetylcholinesterase blockers actually show increased adult-born neuron circuit integration. So this seemed to be like kind of a smoking gun to us. We better dig into the system and see what's happening. So what we did is a genetic experiment where we targeted specifically these cells for ablation by expressing a diphtheria toxin receptor, which is not normally found in mice, treating them with diphtheria toxin and selectively removing only these cell types. Now this is where the whole story changes, and our research program took a pretty dramatic turn. Because away from adult neural stem cells and synapses, we uncovered this morbid obesity phenotype. This was pretty robust. It evolved within weeks of ablating these cells in adult mice. And in fact, these animals, this is a very impressive phenotype. Within weeks, they almost triple their size. And this was correlated to what we observed at first as an increase in food consumption. So this is actually a pretty important thing to test right off the bat. Does this actually have something to do with behavior, or did we just jam the metabolic signature of these animals? So we set up a very simple-minded experiment where we monitored the amount of food that either control or the ablated animals consumed for a period of about a week, just to see how much they would eat. And then we would restrict all the animals to the control animal diet. After that, we'd give them a 2-week period to eat as much as they wanted, and we'd see what would happen. It was pretty obvious that this seemed to be behavioral in nature. The animals immediately would eat a lot. You'd restrict them to the diet, they'd lose body mass and body weight. You'd give them back the food and they'd gorge themselves and immediately become obese again. So this is a good argument for motivation or behavior. So we tested this exhaustively through physiological measurements and also monitoring, of course, blood serum levels, and the upshot that this is a motivated behavior and not a metabolic dysfunction. So this is how we went from neuronal synapses, olfaction, and adult-born neurons into this basal forebrain and addiction feeding circuits. And I think by the time we get to the end of this slide session, you'll see that this isn't just dumb luck, that it's true serendipity, that these are both related. So we decided to step back, and what is the basal forebrain itself? This is actually for a very small structure in humans and in mice. It's pretty complicated. It's a potent source of acetylcholine. That's where we got to it in the first place. Has broad projections throughout the sensory cortices. It's involved with attention, wakefulness, reward. And it also comprises diverse population of neurons other than just cholinergic cells. So a talented MD Ph.D. student in the lab decided to dig into this a little bit further and ask the basic question of how are these cholinergic basal forebrain circuits linked to behavior and feeding. So he set up a very straightforward experiment where he simply took animals and restricted them from eating overnight. Then he would take half of them, feed them, process the brain tissues, and it's called a fast refeeding assay. And what he noted is that those animals that were refed upregulated this immediate early gene called CFOS, which is an indicator of neuronal firing. He also noted that indeed the cholinergic neurons upregulated CFOS, but interestingly noted that another population characterized by the expression of glutamate or glutamatergic projection neurons by the expression of VGLUT2 were actually upregulated more. So this suggested that, in fact, what might be happening here is you're upregulating more of this activity in the potentially a downstream population of cells than these cholinergic neurons. So he took a particular interest in these glutamatergic projection neurons, reasoning that a little bit hot here. The cholinergic neurons only make up less than a quarter of these cells in the basal forebrain. Glutamatergic neurons are over a third of these cells. And he asked specifically, are these cholinergic neurons somehow talking to the glutamatergic projection neurons? Because he reasoned that, in fact, these projection neurons might be the workhorses of pressing this information downstream into targets that are known to be associated with feeding and addiction. So to address this, he set up a cross where he generated animals that expressed this light-activated ion channel rhodopsin, into a background that had a Cree driver in these glutamatergic neurons. This afforded him the ability to simply inject a virus that would label only these glutamatergic neurons with a red fluorescent protein. He could then section the brains of these animals and take whole cell recordings, electrophysiological recordings from the downstream targets while photostimulating the upstream cholinergic neurons. And it was pretty obvious right off the bat that we had robust photoactivated currents. These currents were completely refractory to fast neurotransmitter blockers. So it wasn't glutamate, it wasn't GABA, and it wasn't the muscarinic drive that can be blocked by atropine. You could block all of this firing by treating with tetrodotoxin. You could recover it by treating it with 4-AP. But importantly, out of all of this, it was completely blocked by the nicotinic receptor agonist mecamylamine. So what this said is this is a direct, fast, robust current between these cholinergic neurons to these glutamatergic projection neurons. He next wanted to know what's actually happening in the projection neurons themselves in the presence of food. So we heard yesterday about the Mark Schnitzer approach, and this is what he took by implanting one of these small microendoscopes into the brains of these animals. The basal forebrain is very deep in the animal, so it's an approach that is really good for this type of an assay. And what he did is he introduced selectively into these glutamatergic neurons the calcium, the genetically encoded calcium indicator, GCAMP. So this is a GFP molecule that, when a neuron fires, glows brighter. And we had seen this yesterday a little bit. And in this way, he could monitor the output of these neurons while providing some sort of food stimulus. This is actually a pretty robust assay. He could do this over multiple animals, over multiple good populations of cells. And what he noted is that indeed, in the presence of food or food stimuli, these neurons selectively became active. But this really led to the question, does this have something to do with eating, the presence of food, or could it actually be triggered by the simple perception of food? So Jay set up a pretty clever experiment where we took advantage of our information that we know about the olfactory system, and he set up a passive conditioning experiment where he simply doped mouse chow with this monomolecular odorant called R-limonene. It smells like limonene to us, but importantly, it's inert to a mouse, so it has no valence. It's not considered aversive or repetitive. And then what he would do is for 2 weeks, let them eat the chow. He'd condition it to the odorant, and then he'd simply present the odorant instead of the chow. And compare that to some useful controls. The S-limonene, which is the enantiomeric compound to this, so it's molecularly similar. To us, it smells a little bit different, it smells like a pine tree. But again, to a mouse, this has absolutely no value or valence. And indeed, what he noticed is that he could stimulate these neurons selectively by simply the perception of food. So this really begged the question, how are these neurons actually involved in feeding? Are they driving it, or are they just responding to it? So he took advantage of another little genetic trick where he targeted, once again, the neurons in the basal forebrain, these glutamatergic neurons, for the expression of this bacterial sodium channel called NACBAC. NACBAC, when you express it in neurons, simply opens up the sodium channels a little bit longer than normal, lets in a little bit more current, increases spontaneous firing rate. So in other words, kind of just turns up the volume on these neurons. You don't have to do anything exogenous to them. You're basically just dialing up the activity. And this is where he revealed a pretty remarkable phenotype. These animals, in very short order, within days, voluntarily quit eating. They dramatically lost weight, and indeed this was correlated to decreased food intake. He went on to verify that this is actually Hypo Faeza. This was a voluntary action by assaying the serum hormones, as well as you could actually rescue these animals by liquid gavage. But it's a little bit of a problem when you're assaying these animals when it's such a dramatic phenotype, even though it's pretty compelling. So he had to step back and wanted to ask what's actually happening at the level of the circuit. So he took advantage of the kind of knee-jerk response experiment in neuroscience now by using optogenetic methods, where he implanted a fiber optic to stimulate only these cells. And not surprisingly, he had shown that they decreased their food intake. This is a more acute response. But interestingly, this is where we learned that story takes another turn here, because we've learned along the way that when you simply photostimulate circuits in the brain, some random things can happen to mice especially. Sometimes when you're measuring food, it might not mean that they're decreasing food consumption. They might be having a seizure, for example. So we monitor all of these with video. And what he noticed is that, in fact, this red circle represents a petri dish full of food. If you stimulate these animals, if you don't provide any light but express channel rhodopsin or control GFP, these animals, if you fast them overnight, they'll immediately go to the food. But interestingly, these animals seem to avoid the food. So this was intriguing, but this wasn't a very high-resolution experiment. This could have been just random, but we knew that they were walking around and behaving fairly normally. So he upped the stakes a little bit, and he set it up an experiment where he put food in 3 corners and kept food out of 1 corner. When he would photostimulate these animals, it was pretty obvious. They were avoiding food at all costs. This is even after fasting them overnight, and this is a pretty strong evolutionary drive to eat. So this is quantifiable, and in fact, it was reproducible even with the perceptual cue of the odor stimulant alone. So this suggested that they were actively avoiding the food with the perception of food. So finally, we started with synapses and adult-born neurons, and now we're into this feeding circuit. What is the downstream output of the basal forebrain? How is it taking olfactory information and pipelining that somewhere else? So we set up a genetic tracing experiment that the details of which here don't really matter, other than he could label selectively these cells in the basal forebrain with this marker called synaptophysin GFP that localizes GFP only to axon terminals. So in this way, you could section through a brain, find where the GFP mark resides, and you can identify presumptive downstream targets. And what he found is that there was a very robust projection there was a handful of projections in many places in the brain, but the 2 most robust projections were to this area, the lateral hypothalamus, which was previously described to be involved in feeding control, and moreover, the lateral habenula, which is known to be associated with reward and aversion. We're actively pursuing this right now. So what I've shown you is that we started from sensory cues, brain plasticity mechanisms, what's going on here with perceptual cues. It's drug us headfirst into this basal forebrain region, and it's modulating the cholinergic cells within the basal forebrain, which are associated with attention, wakefulness, and perception. We've identified that indeed they project downstream through fast nicotinic receptors onto these glutamatergic projection neurons, which then pipe out into other areas of the brain to control these almost reflexive behaviors such as feeding and a reward and addiction. We're actively pursuing right now, is this some sort of a convergent line, or are there discrete lines of information going on here? How are these 2 different regions talking to each other? And importantly, the brain has to know what a good sensory input is versus a bad sensory input. So we're mapping the top-down projections and what's telling the cues to be good or bad. And we have indications that, just as a teaser, that it might be involved with the insular cortex. So finally, step back just 1 second from a little bit higher perspective. And we all know that tobacco use changes appetite control. For a long time, it's not known really how this has done this. So we might consider that we have stumbled across this type of brain circuit that is modulating this type of nicotine input, an addictive circuit to modulate appetite. Moreover, if this circuit becomes dysfunctional, could this indeed be a source of almost a reflexive behavior to link certain types of stimuli to addiction or even neuropsychiatric-like illnesses that are associated with eating disorders? So finally, to finish up, I have to thank my lab. They're a fantastic group of people, very generous support throughout not only the lab, but different entities. Great collaboration with Quin-Chun Tong, who has taught us a lot about the feeding circuit. Fantastic place to work. And then finally, I just have to make a brief acknowledgement to the McNair Foundation and Bob McNair, who was the person who basically provided my startup package here at Baylor College of Medicine and to recruit me to Texas. And He's known for being a great businessman and owner of the Texans, but he was actually a pretty insightful human being in general. He really believed in kind of sharing ideas and open collaboration. I think this is one of the huge benefits of the Texas environment. And I'm just going to leave you with a pretty endearing comment that he was documented of saying more often than not, and I think that this is more indicative of kind of a scientific perspective. So sorry for going a little bit over, and thank you for your attention.
We have time for a few questions.
Talk. Curious whether the activation of those basal forebrain glutamatergic neurons causes nausea or malaise, whether it supports a conditioned taste aversion. And as you know, nicotine upon first consumption often causes nausea and might activate that same system in that manner.
Yeah, actually, we haven't tested the definitive nausea, but we don't think it's malaise, and we don't think it's like just a general sickness, because in fact, when you do it with the odor stimulus and you remove food from it completely, they don't even consume the food at all. Once they perceive the notion that food is there, they avoid it. What we don't know is what is telling them that food somehow can be bad. So that's a great question. We addressed that early on. That was our main concern right off the bat. Were we making them sick or metabolically dysfunctional? Thanks. Yes.
Really so interesting. Two quick questions. What are the implications for e-cigarettes and other nicotine delivery devices? Are you looking at that at all?
Yeah, that's kind of a funny question, because with the first obesity phenotype, the first thing we did is we took these animals and we shaved small patches off their back and put Chantix patches on them. And believe it or not, it worked. But the problem is that prolonged nicotine use, as it was alluded to, actually interferes with your whole autonomic system. Your guts get bad, so it hurts their digestive tract. So what you're actually looking at after a little while becomes secondary. But it's very interesting, because either the perception, ways to augment this type of phenotype or change it or alter it, that was one of the first things we did, but it was just difficult to prolong after about a day or two. We could change their diet, but it's a great question.
And actually, my second question is for Dan. I left my computer and charger on the podium. Could you hand it to me, please?
All right, thank you.